Welcome to the Ag Emerge podcast, brought to you by Ag Solutions Network. Your farming challenges are unique, so your practices should be too. We're here to share emerging ideas, build connections, and provoke conversation.
Get ready to improve your soil, your crops, your livestock, and your family's livelihood. I'm your producer, Kim Chase.
And I'm your host, Monty Bottens. Thanks for joining us.
Thanks for joining us. We are excited to bring you this special podcast. It's the live recording from the very first Ag Emerge event that was held in Monterey, California in 2019. It all began when Monty read a paper Dr. White had written about the rhizophagy cycle, and he knew he needed to get this powerful research and information in the hands of farmers and ranchers. Dr. White had everyone on the edge of their seats as he shared this groundbreaking research. We've included Dr. White's main stage presentation along with his breakout session. But first, we bring you a special clip of Monty and Gabe Brown discussing what they heard, because Gabe was both a presenter and in the audience that day when Dr. White was on stage. Enjoy.
One of the fun people that you met at Ag Emerge was Dr. James White.
Yeah.
And
I was just going to
look where that's gone from there.
I was just going to say that, Monty. I give you credit because I had not heard of Dr. White. And I'll never forget, I listened to him. I walked out of that room when he was done speaking. I immediately called Shane and Alan. And I said, hey, there's a guy. You got to hear this guy. This is phenomenal. And to your credit, I believe that was the first ag conference Dr. White had spoken at. And I'm like, how did we not know about this guy? And that has been just taking soil microbiology to another level now, that interaction between plants and biology. I mean, it's just fantastic.
Yeah,
the work that he's doing.
Mind-blowing. And that was one of the things we hope to do here with Ag Emerge, is find those people that are doing unique work that just don't have that platform or that voice or those technologies that just, that can benefit regenerative agriculture and bring it into the discussion. I want to take just a quick minute to introduce our first of our Ag Emerge Talk speakers, Dr. James White. He's from Rutgers University. I get a chance to read a lot of research papers in the industry, and I ran across his paper, and it just blew my mind. So I'm really looking forward to you hearing from Dr. White and what he's discovered, and as he kicks off our first of our Ag Emerge Talks. Dr. James White.
Thank you, Monty. I'm very happy to be here and to talk about this research to you. What I'm going to talk about is something that we've termed the rhizophagy cycle. And this is a process whereby plants can extract nutrients from soil microbes. And so these microbes actually are endophytes. Okay? And endophytes are microbes that enter into the tissues of plants. They can go into the shoots of plants, into the leaves, they go through the seeds in many cases, they go into the roots. And so what we've been looking for is how these endophytes might be affecting plants. And they can be fungi, like the image I showed before, or they can be bacteria. And this is a root hair of a plant. And you can see these recently divided pairs of bacteria actually inside those root hairs in the root cell. But they're inside the cell, but they're not inside the cell because they are outside the plasma membrane, but inside the cell wall. So they're in what we call the periplasmic space. Endophytes are everywhere. All plants have endophytes. In fact, they have multiple endophytes. They have fungi and they have multiple species of bacteria. Okay, so there's a small community inside plants. And they have major effects on the plants. Okay, for example, and what I'll be talking most about is the nutrition absorption function of endophytes, but they They also will modulate development of the plant. Okay, the plants don't grow right without these microbes inside them. They will improve the stress tolerance of plants. When the endophytes are there, plants have more tolerance to oxidative stresses. That's abiotic and biotic stress. And they will suppress plant pathogens. Okay, these endophytes, when we're on a seed, for example, seed germinates, they'll go out into the soil, they'll colonize the pathogens, and they will reduce the growth and the virulence of those pathogens. So there are all these beneficial effects that these microbes are having for plants. Plants evolved to have these microbes with them. Okay, and finally, they will suppress competitor plant growth. Okay, so this is something we're actively exploring now, and I'll mention a little bit about it at the end of this presentation. In 2010, some investigators in Australia discovered that microbes, yeasts, and bacteria were entering into plant roots. And they actually labeled the microbes with green fluorescent protein so they could track them inside the plant. And they took these images and they showed that these microbes went inside the plant cells. And they envisioned this as a root eating process, that the roots were actually consuming these microbes. And so they denominated this rhizophagy, root phagy for eating, rhizophagy. So they published an article titled "Turning the Table, Plants Consume Microbes as a Source of Nutrients." Well, we've been examining that phenomenon for a few years now, several years. And in order to visualize these microbes in the plant, we developed a new staining procedure. And basically, it's a reactive oxygen staining technique. We use a special chemical, DAB, but it will stain brown wherever reactive oxygen is present. And so you can see this seedlings in agarose, in agar here, and that brown, this has been stained. The brown is where reactive oxygen is present. So what happens is, when these microbes are in the plant reacts by secreting reactive oxygen onto them. And so we're able to see these microbes, whereas before these microbes were almost invisible. Now we can actually visualize them with this technique. So that's what we've been doing. And what we have found is that these microbes actually, they alternate between being endophytes in the plant and going out into the soil. So they will alternate from soil to enter the plant actually at the meristem, root meristem tip. They go inside the cells at the meristem where the walls are soft. And they actually will convert to protoplast phases. They lose their cell walls. They form protoplasts. And eventually they will trigger root hair formation. Okay, without the microbes, we get no root hair formation. The microbes are necessary to start that process. Then, as the root hairs elongate, the microbes are ejected out of the tips of these root hairs. And they go back into the soil, reform their cell walls, go out, gather nutrients in the soil. Then they're attracted back again to the meristem. This is the root exudate zone. And enter the tissue again. So this cycle continues. This image shows bacteria as they enter root cells. They're just inside the root cells there. Okay, this image shows fluorescent microscopy, shows the bacteria exiting the root hair tip. Okay, so this is Phragmites, invasive reed grass, one of the plants we work on. And you can see the exudate zone here, this cloud of microbes, all these blue dots. And then this zone here, right around that is the meristem where the bacteria, in this case, are entering those plant cells. Exactly what triggers them to enter, we don't know. There's a couple of hypotheses out there. I won't go into the details of that. but they go inside there. Once inside, the bacteria will shed their cell walls, and they form these protoplasts. And these protoplasts actually don't function like regular cells. These protoplasts will divide kind of like yeasts, but by blebbing. They'll just bleb out. Normally, bacteria with cell walls will have to form, they'll do binary fission, they divide, and they make a cell wall. And so it's a very slow process with normal bacterial division. But without cell walls, they just bleb out into smaller and smaller sizes. And so this internal, this actually shows some of these L forms. Actually, this is in culture. And you can see they just bud like that sequentially. So in this phase, the plant essentially can replicate very rapidly these microbes. And so this actually is one of the things that happens is these microbes are replicated inside the plant cell. But at the same time, reactive oxygen is bombarded onto these microbes. And these actually show, this is a parenchyma cell. You can see these little red cells. These are the protoplasts of bacteria. And the red is from the reactive oxygen. And the form of reactive oxygen that's used and is highly potent is called superoxide. So superoxide will penetrate into the membrane of the bacteria, will go into the cell, will break down some of the proteins, and the older protoplasts will actually degrade. And you can see actually, here's one, you can see this bigger one that's clear, then the smaller one that's blue. This is stained for protein using aniline blue. And so the smaller ones have a lot of protein in them, these larger ones don't. Okay, so this reactive oxygen is very effective at extracting nutrients from these microbes, while at the same time propagating them, making them so they have more numbers. So the plant is going to push these microbes out back into the soil, increasing the numbers of microbes that are carrying nutrients for the plant. So we also have found that fungi do the same rhizophagy cycle. They go inside the cells, and this is one of the plants that, this is actually a little weed called Frolichia, snake cotton, Frolichia gracilis. But a fungus will go into the cells in this particular plant. Okay, and the fungus is this yeast. Okay, this is called a black yeast. Okay, these fungi, once they enter into the plant cell, the root cell, they lose cell walls also. They form protoplasts. Okay, and this shows these protoplasts of fungi forming. And these protoplasts, actually, they call these mycosomes, the fungal protoplasts. But it's the same thing. It works exactly the same way as the bacterial. Okay, this shows the protoplasts inside Frolichia. You can see there's chains of them, again in the periplasmic space outside the membrane. And you can see here, the same plant, you see bacteria down in here. So there are both fungi and bacteria that go into these cells. So it's a mixed community. The rhizophagy microbes modulate development of the plant. Okay, and for example, roots don't show the gravitropic response without these microbes being there. No microbes, roots grow on the surface of the soil, or they'll go into the air in some cases. When the microbes are present, roots will grow down into the soil. So the plant is waiting for those microbes to trigger it for soil penetration. Also, root hairs. Roots don't develop root hairs without these microbes in the cells. Okay, if there's no microbes there, you get no root hairs at all. This shows a Bermuda grass seedling, and you can see the tip here, and a little bit further on the root here. And there are no microbes here, there are no hairs here. Okay, and the same experiment with pseudomonads. They go into the tissue, and immediately hairs form. And what's happening is, of course, the microbes are going in here, they're triggering hair development, and they're coming out. function of hairs. We all know hairs on plants, root hairs on plants are to absorb nutrients, but maybe not. With the rhizophagy cycle, it's clear that these hairs, this is a root hair here, is the vehicle for putting those microbes out into the soil, putting them back into the soil to acquire more nutrients. Now you can see the hairs, this is fluorescent stain, you can see all the bacteria. This is a close-up of a hair, of actually Poa annua. Little dots there, those are the protoplasts in there. And you can see coming out here, these are the protoplasts being extruded from the tip of the hair. So putting those microbes back out into the soil is necessary so that they can then go out, get new nutrients, and then carry those back to the root at the growing tip and re-enter. Okay, this shows a hair. This is a sedge, and you can see this is the hair. These are all the protoplasts inside it, loaded with protoplasts, and these actually are in motion. There's cyclosis that's moving these microbes around, so they're flowing like this inside these hairs, and we have images that I'll show tomorrow of that process, and these are the bacteria being extruded out. Once they're pushed out of the tip of that hair, they reform their cell walls. You can see there's rods out there. Okay, this is tomato that shows the bacteria being extruded, and bacteria, blue staining here and some bacteria there. First of all, what nutrients do microbes get for plants? Nitrogen. We've shown 30% of the nitrogen comes from these microbes, but we also, there's good evidence that they're minerals, iron, zinc, copper, other minerals, those minerals that plants need. Microbes are very efficient at getting those minerals. First of all, they can swim out there, they can find the mineral, they can latch onto it, and then absorb it, and then bring it back to the plant. So it makes all the sense in the world that plants would learn how to manage microbes in order to obtain nutrients. Okay, and how do they do that? They do that with structures called siderophores. They will also, simply their cell walls absorb nutrients onto the cell walls. And so when plants take those cell walls, they get those nutrients. And plants use organic acids to kind of release some of those nutrients, and the microbes also will go out and get those immobilized, complexed nutrients like iron, citrate complex, and absorb those. Okay, so first of all, siderophores, they're structures, they're molecules that microbes have, like this is a siderophore, that will actually go out and hook onto, for example, iron. And then this will then mobilize back and hook into the bacterium. So it's a specialized structure for acquiring metals, for example. Okay, this shows that actually the cell walls themselves of a microbe has a net negative charge. These metals have a net positive charge. So the metals will just adhere to those cell walls. And this actually shows silver particles in a cell wall of a bacterium. Plants actively secrete organic acids, like, for example, citrate, malate, acetate. They'll secrete those into the soil, these exudates. Those organic acids will then complex onto these metals, like iron, form a complex all over them. Microbes have special organic acid metal transporters in their walls and their membranes. And so these microbes can get these complex nutrients, organic acid metal complexes, absorb them in. Okay, so this is a case where the plant is working with the microbes, secreting all these organic acids out there to immobilize the nutrients, sending those microbes out to then gather those nutrients for the plant and bring them back. So we have also discovered that we can use these rhizophagy microbes to stimulate development of crop plants, but competitor plants, we can suppress that development. That is, it'll make once some plants grow and other plants, they'll suppress. And so we're trying to develop bioherbicides, growth promoters that are also bioherbicides using that. And one example is a tomato endophyte. We got an endophyte from tomato, it's seed vectored endophyte. It happens to be this, Micrococcus luteus. You see it in the root hair there. It stimulates tomato development, but it inhibits the growth of, for example, in this case, carrot. Here's with carrot without, here's carrot with. You see all the little white areas around there, all these white zones? Okay, those are the microbes around the seedling. Okay, and this is Japanese knotweed. Okay, this is without the microbe, this is with the microbe. And you see it suppressed Japanese knotweed completely. Okay, so just to summarize very quickly here, these endophytes and this rhizophagy cycle has some ramifications for agriculture. And one of the ramifications that is like no-till is better because you don't disturb that microbial community. Organics in the soil is better. Because these are seed transmitted, you don't want to have sterilized seeds. You don't want to have special seed treatments that remove the microbes and put something else on it. Okay, so you want to preserve those microbes. And finally, in the future, based on the work that a lot of people are doing, we might be able to use microbes to manage crops without agrochemicals. So that's it. Thank you very much.
The AgEmerge podcast is brought to you by Ag Solutions Network. The ASN team is hands-on, digging in and invested in regenerative agriculture. Along with the proper plant nutrition and biologicals to boost your soil microbiome, we provide the ideas and implementation guidance to support you on your soil health journey. So stop farming the same way and contact Ag Solutions Network today at asn.farm.
So the process is called the rhizophagy cycle, rhizophagy, root, phagy, eating, and cycle because it's a cyclic process, and I'll talk about that. And it's through this process that plants actually cultivate microbes and then extract nutrients from the microbes. And these microbes are endophytes. An endophyte is a fungus or a bacterium that goes inside plant tissues. And so mostly, if you're familiar with fescue grass, you know there's fungal endophytes in fescue grass. Okay, so this actually is an image that shows one of these fungi in fescue grass. But actually, there's all kinds of endophytes in all plants, fungi and bacteria. So every single plant has endophytes in it, and usually it's multiple endophytes, like small community of endophytes. Okay, and some of the features of these microbes in plants is that all plants have them, fungi and bacteria. Intracellular, that actually means they go inside the plant cells. And intercellular, they go between the plant cells. So they're both, in both cases. They will improve stress tolerance, abiotic stress tolerance, so they make the plants more resistant to, for example, drought, heat, heavy metals, anything that kind of relates to oxidative stress, they'll protect the plant from. So they're very important to be in plants for partially that stress protection. You have a weak plant without any endophytes. Weak, poorly developed, they suppress plant pathogenic fungi. Okay, we know that some of these endophytes will go out of the plant, go out into the soil, and colonize these fungi that are out there, these pathogens. And they don't necessarily kill those fungi, but they change the aggressiveness of those fungi. They'll make a fungus that's pathogenic, they'll turn it into a non-pathogen, simply saprophyte or even another endophyte in some cases. Exactly how they do that, we don't know. They will modulate plant development. They will actually control the development of plants, especially the roots. We see that kind of control by these microbes. they improve nutrient absorption. And this is one of the main processes that we'll talk about. So this is one of the nice things about doing work on plants is plants are all over the world, and so I get to travel to those places and collect and work on those plants. Okay, so this is actually the island of Bonaire and the Dutch Antilles, and they have these huge cacti. They call them Kadushi cactus, all over this desert island. And the fruits of these cacti are up here. But if you extract from those fruits the seeds, okay, that's shown here, and then germinate those seeds on, say, agarose, something clean, so that you don't have too many other contaminants coming in, you get a seedling that looks like this. And this is actually stained for reactive oxygen. This is stained, and you can see this orange root. Okay, so it's turning orange because of bacteria that are inside the plant. And what we do is we'll use this, I call it reactive oxygen staining, but it's a chemical that will turn brown if there's reactive oxygen. Reactive oxygen, hydrogen peroxide is one. Superoxide is a very powerful form of reactive oxygen. So the plants actually secrete superoxide onto these microbes. And so we're using this staining technique because that allows us to see those microbes. If we didn't have this, those microbes would be invisible. Okay, and it's because of that that people haven't discovered this process before, because nobody could see those microbes in the plants. But a few years ago, we developed this technique, and you get this brown coloration using this technique. The stain, by the way, is a fancy diaminobenzidine tetrahydrochloride that we use. We just call it DAB. That's it. Okay, so this shows that root of the cactus, the Kadushi cactus. This is actually a root hair. These are root hairs here. And you can see inside. You can see all of these. Those are all bacterial endophytes. Those root hairs are loaded. Other root cells are loaded with these endophytes. And this shows a close-up. And you can see root hair. And you can see these divided pairs of bacteria there where they recently divided. Okay, so you can conclude that these microbes are replicating inside this plant. Okay, now they're actually not inside the cell itself. They're actually outside the membrane, the cell membrane, but they're underneath the cell wall. So this line there shows the cell wall. So they're really between the cell membrane of the root and the cell wall. So they get in that space called the periplasmic space between the membrane and the cell wall. In 2010, some investigators discovered that plants were internalizing microbes, yeasts and bacteria. And they actually, they wrote a paper called Turning the Table, Plants Consume Microbes as a Source of Nutrients. And later they named this process rhizophagy, root eating. Okay, so what they did actually is they labeled their bacteria using a fluorescent protein, and then they visualized it. So you can see here the bacteria or the yeast, I don't remember which one this is, inside the cells. Here you see them inside the hairs. Okay, and so they actually went on to start a company where they're from Australia. They started a company where they're using yeasts as fertilizer. Okay, so they're trying to monetize that discovery. We discovered actually in some work that has been going on over the past 8 years or so, we've discovered that the process is actually cyclic. And we published an article just a few months ago in the journal Microorganisms, Rhizophagy Cycle, an oxidative process in plants for nutrient extraction from symbiotic microbes. So we basically described this process in more detail that we've been able to ascertain over these 8 years. And the process is like this. Okay, this is a diagram of the root. The microbes from the soil actually are attracted over to this root tip where these cells are dividing, and they grow in a zone out here, an exudate zone, just outside that meristem. And what happens is the bacteria or fungi become internalized into these root cells. And when they're internalized, they will lose their cell wall, and as they're hit with reactive oxygen, the superoxide that I talked about, the plant secreting. So that causes them to lose their cell walls, they form protoplasts, and then they will eventually trigger root hair formation. They trigger the hairs to form, because without the microbes, we don't get any hairs forming at all. And then they leave as they will exit the tip of these hairs as they elongate. They're actually, and I'll describe that process, they're actually ejected from the hairs by the plant. And they go back out into the soil, and then there they will acquire nutrients and carry those back to this exudate zone, and some of them will re-enter again. So this cyclic process. So they acquire nutrients in the soil, nutrients are extracted inside the cells of the plant, of the root cells. Okay, and this little image that shows a root hair, and you can see the bacteria there. There's fluorescent stained little cloud of bacteria coming out of that, being ejected out. This is a cell near the meristem, and you can see this is the cell here, the root cell. You can see the bacteria inside. So a lot of these microbes, this is a grass seed, this is a typical grass. I think this is a poa of some sort, bluegrass. These grasses will carry many of these microbes actually on the surface of the seed. They have lemmas and so forth. They have tissues that adhere to the seed. So they're adapted to vector. Some of these microbes are actually already in the embryo or inside the seed. So they have some surface, some interior. Different groups go different ways. But here you can see a germinating tall fescue seed. You can see the root coming out. See this kind of yellow around? These are the microbes that came off of that seed. So they're colonizing that seedling. They're colonizing at the shoot part, and they're colonizing at the root part. So the initial colonization of the root is right at that meristem. And you can see these diagrams indicate this where the root exudates are. And so the microbes grow right there initially. Here's another one, shows those exudates there, and that's where the microbes grow. This is an image that's an invasive reed grass, the giant Phragmites that's invasive in the United States. But this is the root, and here you can see the exudate zone here, and you see all these little dots. These are all the microbes. And what happens is the microbes enter right there, right where the cells are dividing, and they have thin walls, and the microbes go inside there. We don't really know how they go in. We don't know what process it is. We don't know. One theory is that the plant cells that engulf them. You know, there's another idea that the plant is manipulating the microbes and causing them to enter like a parasite would enter. So we don't really understand that process. All we do know is they do go in. So this is just a close-up of that. And, entry into these cells would be right in this area where you see this irregular surface there. The root tips actually show, even without the microbes, show high production of this reactive oxygen. This brown area shows where a lot of reactive oxygen is produced. We think that it secretes that all the time there. They're secreting the reactive oxygen, probably because as the microbes enter, they're going to be converted to protoplasts right away. They're going to be hit with reactive oxygen. Once they go in and they're hit with reactive oxygen, they lose their cell walls. Okay, and they form protoplasts. There's naked cells without cell walls. And there's actually, this shows an electron microscope image of some of these protoplasts of bacteria. And you can see these things just like bud out in little chains. And there's small ones, little tiny ones, and there's larger ones. So their rules of these cells aren't the same. when they're in the protoplast stage. That's when they're in bacteria stage. Bacteria, you measure bacteria, they'll be very similar sizes, but with their cell walls. But with these protoplast stages, they could be really, really tiny, or they could be bigger, and there's highly variable. So that's an important difference. These also replicate very rapidly. So once they're inside and they lose their cell walls, the plant can replicate them and make lots of little copies and then send all those copies back out into the soil to get more nutrients. So this is very important for that in this rhizophagia. Okay, some of the work I mentioned we did with giant reed grass. This actually is the root of giant reed grass. You can see this is a cell, a root cell here. And you can see these little blue or reddish brown structures. These are those protoplasts. And you can see there's a chain of them here. You can see this is the oldest one. You can see it's clear, larger and clear. And this smaller one is bluish. We stained it for protein using a protein stain that turns it blue. And so you can see these smaller ones have lots of protein in them. These older ones don't. What happens with reactive oxygen, it will go into the cell, make the membrane leak, cut up some proteins, and those proteins leak out, and so forth. So it's actually cell becomes leaky. And some of these older ones will just disappear, and you'll have only the younger ones that come out. So in order to make reactive oxygen, the plant actually uses oxygen. Takes oxygen from the atmosphere. And they use the enzyme. This is the enzyme actually in the membrane of the root. And they take that oxygen from the atmosphere and they make superoxide. So that's what's going on, just so you see a little bit of the chemistry. This actually shows a lot of these little protoplasts inside a root cell. You can see big ones and then you see smaller ones here, although it's very hard to see. So these cells get loaded with these protoplasts, microprotoplasts, and they degrade. Some of them degrade completely. Okay, this shows a little bit better. You see younger ones here, they're blue staining. You see the older ones, they're bigger and clear. So many of them degrade completely. So they're just getting nutrients out of these. We used to think it was only bacteria that did this, but we discovered a couple of years ago or a year ago, something like that fungi also do this. And one plant that does it is this plant. This is a snake cotton. It's amaranth family, and a number of species in the amaranth family will use fungi instead of or in addition to bacteria. And this is one of the fungi. It's a yeast called a black yeast. These fungi will enter into the tissues at the root tip meristem also, and they will then turn into protoplasts again. And they form protoplasts that behave just like the bacterial protoplasts. They just bud like this inside in chains, many chains, and they go in the same place. They go into the periplasmic space in the root cell. Okay, so they're located in the same place. They call these mycosomes, though. A fancy mycological term that, of course, mycologists who studied this had to give a special term for it. But it's just a protoplast. Just a protoplast. This shows it in that plant, in the snake cotton. This is a root parenchyma, and you see these protoplasts in here. This is the fungus. But you see also here, in the same root, you see these. These are bacteria. So the plant has both fungi and bacteria in it. So you have these communities. That's just visual evidence that you have more than one kind. Plants require these fungi and bacteria, these microbes, in order to develop properly. If you take them away, they don't develop right. They won't grow right. And for example, plants without these microbes don't show the root gravitropic response. Okay, so if there's no microbes there, roots will grow into the air, stick right up in the air, or they'll just grow on the surface. But they don't grow down into the soil. They just lie on the surface, perhaps waiting for the microbes to come. Also, roots without microbes, plants without microbes, don't show root hair elongation. they'll make roots but no root hairs. Okay, so those microbes are necessary to develop root hairs on the plant. And this shows a Bermuda grass seedling, and you can see the tip of the root here, and a little bit older part of the root here. And this is without microbes. All the microbes have been removed. There's no root hairs forming on these plants. Okay, with microbes, we put a pseudomoniad on it, which we got as an endophyte off of another plant, and we put it on, and you can see root hairs form immediately. And you can also tell maybe that there's brown in here. That's where the microbes are located. Okay, and when they first go in, when they go into the root cell,
When you say immediately, like 1 day, 1 minute, 1 second, on the root here, like how fast is that time?
days. It's a couple of days. In order, it goes in, and then the hairs begin to form, right? And they'll form over a period. It probably depends on the plant. So we're talking about maybe 2-3 days, something like that, before you get full elongation of the root hair. Okay, so this is actually what happens when they first go in. They have their cell walls. When they first go into the root cell at the meristem, they have this. That's the bacteria, the pseudomonads with the cell wall. And you can see in here some others. These are pseudomonads with the cell walls, but they immediately will lose those cell walls once they're hit with reactive oxygen. Okay, this is part of the root, and you can see. Actually, you look at this root, you see all these brown dots in there. Those are all the microbes inside those root cells. And you see these brown dots out here. Those are all the microbes inside the root hairs. So there's lots of microbes in those roots. Okay, this is a close-up of a root hair, and you can see the microbe protoplasts actually in there, the brown staining. And that's from a Bermuda grass seedling. This is a Rumex crispus curly dock. Okay, we use that as one of our lab guinea pigs, and we isolated a Micrococcus luteus that I mentioned it yesterday from tomato, but it's distinctive because it forms these little tetrads of cells. And so you see, and the tetrads are distinctive enough, we can tell when they disappear. So this is around the meristem, and you can see the tetrads that have just entered, and they're inside those root cells there, at least that outer layer of cells. This is without any microbes on it. You can see the same meristematic cells free of endophytes. This is a close-up of that. So you can see these tetrads are these little packets of bacterial cells. They all still have their cell walls, but they're all inside those root cells. Immediately, they will lose the wall. They will lose their packets of cells. They'll separate out, and they'll form the little protoplasts. And here you can see them now. They're the same bacterium, but it's now forming little spherical structures. It's a close-up of that, and what you can see is that if they're actually out in this periplasmic space, they're out here, just outside. But there's the wall, there's a membrane there, and they're out, located outside of that, so you can see the position. Okay, just similar thing. You see the microbes, the protoplasts inside the hairs, protoplasts inside the hair there. Okay, some of these will spontaneously turn back to walled cells inside the plant cell. So you see, here's our Micrococcus. This is from culture. Micrococcus luteus bacterium forming the packets. You see, most of these are little bitty dots there. Those are the protoplasts that are still in protoplast phase. But you do have some, like here, here, and here, that are reforming their cell walls and reforming their packets. So some of them spontaneously will go back. The plant will multiply the microbe in the root hair and increasing the numbers, and then eject it out into the soil. And this actually is a root of a grass, and it shows how these root hairs are elongating out away from the root surface. It would not be a good strategy to just put the microbes right there on the surface. It's better, more efficient to move them out into the soil and then squirt them out there. Yeah.
Quick question. You said that they populate inside of the root. before they are exuded back out. Do you have any estimation in terms of the population increase inside of the root?
No, we really have no numbers, but we can see them replicating and I'll show you images that show that. And also the numbers that go in, if you consider
It's like a
entering
ratio.
one cell, you may have a dozen of them, but coming out, you have many, many more.
That was my question.
Yeah, you have many, many more. I can't tell you.
Like
It's
thousands
likely what's
more,
going on is that the plant has good microbes and it's replicating the good ones and then pushing them out into the soil to populate the soil with the good ones so that they can go get nutrients for the plant.
Why do you think then some of these are rebuilding their cell walls within there? I mean, they're
That's a very good question. That's exactly right. They can't go out. If they have cell walls inside, they can't go back out. Okay, so what's happening is Micrococcus luteus is very oxidatively resistant. And so it's able to resist the degradation that you'd be seeing like Pseudomonas. Oxidatively resistant, what I mean is it makes antioxidants, right? And so the plant is squirting superoxide on it, it's making antioxidants, and so it's fighting that process. And it's, in a sense, it's not adapted, especially with this curly dock that we put in, it's not adapted to curly dock. And so it's not optimal association. The function of those root hairs is to put those microbes out into the soil. And this shows root hairs, and you can see this is fluorescent stain. You can see that all these clusters of microbes that are coming out. There's just spewing these microbes out of these hairs. You know, there's a lot that are actually coming out. This is a Poa annua grass. You can see the little dots in here. Those are the protoplasts in there, and they are ejected out the tips of these, and you can see bacteria here reforming. These are protoplasts that came out. You can see almost there looks like a rod there, but they reform their cell walls once they're outside. And the plant actually feeds them out there. It gives more nutrients so that they can increase in size. So this actually shows tomato root hair tip. You can see these microbes in it. You can see a stain for reactive oxygen. See they're all brown, so they get hit with reactive oxygen there. And you see on the surface, you see some little rods forming. Okay, so the model, the way that we think this works is, there is cyclosis circulating cytoplasm here that goes around like this, goes all around the cell, constantly circulating. And that drags these microbes along this rigid wall here. And this tip is thin walled, very elastic, and it just deposits them there at that tip, and they accumulate there. And then they stimulate as this protoplast, this root hair protoplast, pushes, elongates more, and pushes them, puts pressure on them, and then they squirt out little pores that form there as protoplasts. The protoplasts squeeze out the pores, then they reform their walls. Okay, so we'll show, I have some images that we'll look at that. This is a little sedge, again from Bonaire. It occurs in these limestone rocks. There's hardly any soil there, but it's a cool plant to work on. Okay, this actually is that sedge root hair. And root hair right there. You can see in here the microbes out there, right there. And I'll show you another image that shows that movement of the cytoplasm like that, dragging those microbes around. Dragging them around, roughing them up, causing them to split apart and make smaller protoplasts. And okay, so here this shows actually some replication in those hairs. See the hair? You see the protoplast here, the different sizes, smaller ones there. Okay, so those clusters are moving around, breaking up, and so forth. Okay, this is that sedge hair. You see it's swollen at the tip. You can see all those protoplasts in it. You see protoplasts here. Actually here, their wall cells are coming out. The bacteria are coming out of that in a mass from that tip. They're being pressed out, pushed out. These bacteria go throughout the plant, and they're seed transmitted, so they actually are also colonizing that ovary tissue, so that when that seed is fully formed, it has some of these microbes on it. So yes, how significant, it's obviously significant. Which ones do some not do that? Yes, some don't, probably don't do that, but some are seed transmitted, so they're going through the plant. Okay, so this is one movie of that sedge root hair, and what you see is the root hair here, and you can see the microbes here, clusters of microbes moving around, moving like this. But you can also see the tip where those clusters of microbes are locating. And so they're being jumbled around, and as that protoplast here that goes into there elongates more, it pushes them through little pores there at the tip.
Is that real time or is that time lapse?
This is very close. This is probably double speed, but yeah, some of these are moving pretty fast. I mean, we've estimated a time 8 to 11 micrometers per second. So that's pretty fast, really. So how do we do this, right? How do we do this? We grew these in agarose, these plants in agarose, and we turned the plate upside down to visualize it with the microscope, and then we did the photography that way. It's very close to real time. You can visually see them moving. So it's not really time lapse. To speed it up a little bit.
So I understand circulatory systems within me. I'm no expert, but I understand I have a heart that provides the fluidic force to circulate. Within a plant, what is actually causing the motion of fluid?
So plant cells have an internal cytoskeleton. So they will use that cytoskeleton. It's made of filaments and tubules and things. And so they can move those, short them, make them long, and to make the cytoplasm like that move. So this is a circulatory system in plants. And, we can guess why they move like that. The function here is probably to drag those microbes to the tip so it can shoot them out. But it also may be to get nutrient exchange between the microbes and the plant, because you don't have a gradient developing there. You're constantly breaking that down so it's the fastest possible transfer. Okay, so that may be the function. We're guessing at it, but it's logical.
And you're also having two-way traffic within the same tubule.
You can have two-way traffic. Yeah. Typically, I describe here where it's, you can see this. Here's another one where a pore is going to form here, and it looks like you have multiple traffic, multiple ways. It's not random. It's not random. Yeah, it's not random. No, plants are doing this. Plants are really doing this. We can see it. And they're ejected through pores. I don't know that we see an ejection right here, but you see a pore forming. So these pores form and they go out. Okay, this actually is a fescue grass, and you can see the root hairs here. You can see all this. This is sped up a bit, so it's really cooking. This one's really cooking. But you can see a protoplast being squirted out of one of those. At least we think that's what we're seeing there. We're trying to get better and better images. I have an undergraduate working on this now. We think that what happens with the root hairs is they will grow until all the microbes are gone, and then they stop growing. They'll just stop. Okay, if there are fewer microbes, you have short root hairs. If there are many microbes, you get big root hairs. And we think what's going on is at that tip, the microbes are secreting nitrous oxide, or it could even be ethylene, but most likely it's nitrous oxide. And that nitrous oxide stimulates the root hair to elongate. So as long as the microbe is there, nitric oxide is formed and the hair elongates like that. Okay, so that's what we think. At least that may be the chemical mechanism. Okay, so tomato, look at tomato, development in tomato, these root hairs, these root cells will form these little papillae on them. And that's the precursor to the root hair tip, actually, precursor to the tip. And those papillae then will accumulate the microbes. You can see a papilla there, you can see the microbes in it. See, there's not very many there to begin with. Okay, the papillae enlarges, you can see more microbes in there. And here you see the microbes there. You see here's an older root hair of tomato. And you can see how many microbes now at the tip, at that papilla, at the very tip. And of course the wall here is very thin and elastic. And it's breaking, as we said, making little pores in it, expanding. This shows the tomato again. You can see how expanded it is with all these microbes in it. And there you see another one. And there you see another one, microbes there. Here you see the expulsion of protoplasts from the tip. You see those protoplasts accumulating out there. Okay. This is tomato again, and you can see all those microbes there. You can see the microbes on the outside and some just almost being extruded there. English ivy, these have roots that go into the air, so it's easy to see them. These are some of the roots, some of the air roots, but you can see here, English ivy, this is a root hair. You can see microbes there at the tip, and you can see the little pores right there forming there. It's a close-up of that. You can see those little pores that develop, and the microbes are the protoplast is squirted through that. This shows English ivy hair again, and you can see the protoplasts accumulating on the surface there as they're pushed out. Yeah.
I have a question.
Yeah.
As they're expelled out, is there something that they absorb from this the root surface to attract certain metals? Like, for example, if there's like a zinc deficiency, do they have certain things on the surface to chelate that?
Yeah, we'll talk about that. Yes. So we've been putting thought into what the mechanism, how they might be working, and I'll go into that very shortly, and how we think that's happening. Okay, it just shows more protoplastic tip, and this just shows more of that. You know, to get the point. You can see the pores there. You see the bacteria reforming their cell walls here. You can see they're in tetrads here, and they just spill off that tip. You can also see the channels through the cell wall there at the tip, and the bacteria reforming right there. And underneath these spherical protoplasts, okay, you just show the spilling off there. Okay, and this is actually the fungus when they come out. This is a fungus, a yeast again. Rhodotorula is its name. But you see the protoplast inside, and this is actually in clover. This is clover, so clover does it too. And you see those protoplasts. You can see protoplasts there. You can see them coming out. You can see the youngest ones are smaller. Oldest ones out here are bigger. And so what has to be happening is here, these yeasts are getting nutrients as they're being produced outside, suggesting that the flow of nutrients actually is out of those hairs, going to the microbes as they reform their. And what nutrients? Carbohydrates, probably. That's what's cheap for the plant. It can make lots of photosynthate. Okay, this just shows that again. In clover again, you see the formation of the. Okay, nutrient absorption function, how it works.
So the root hairs, are they ever pulling in?
We don't
from the
know.
soil, or is it only?
We don't know. We don't know the answer. We can't say really. One thing is for sure, root hairs die very shortly after all these microbes are ejected. So do they really absorb micronutrients from the soil? We don't know. We don't know. I can't say no, but it certainly looks like their function is to put the microbes in the soil rather than absorb nutrients from the microbes, from the soil.
Can we get a refund on our textbooks?
I know, me too. All the way back to like second grade. Yeah, if it's true. But okay, so to test this, we did a nutrient, to test to see how these microbes were assisting in nutrient absorption. We did an isotope tracking experiment. This is actually where we labeled some protein with isotopic protein, nitrogen 15 it's called, so we could track it, and as it goes into the plant. And basically we put the protein, labeled protein, into agarose here, mixed it in there, and then we put the plants with microbes here, bacteria abundant, okay, and without over here. And then we measured the amount of isotopic nitrogen that went into each of these seedlings. And what we saw when we did that is that when the microbes are present, you get this much. When the microbes are absent, you get this much. And so that is about a 30% difference. So it looks like in terms of nitrogen, the microbes are providing 30%, they're worth 30% of the nitrogen that the plant gets. That's not 100%, but it's significant increase in nitrogen. Okay, so this is a plant grown with all the microbes present, and you can see it's a nice green plant, they're big, lots of roots. And then we take the microbes away, you see now we've got yellowed plants. Then when we put on one of our endophytic bacteria, we reacquire root development here, and they're green again. And here also we have a bacillus, an endophyte that took off of these plants, and then we put them back onto sterilized plants. And you can see that the bacillus also is reacquiring that growth, the green again, of course, lots of root development. Okay, so how do microbes do this? Okay, so microbes have structures called siderophores. They have some other, these are actually iron molecules, is how they get iron. Cell walls will absorb metals to them. They also will actually work with plants and getting microbes using from the organic acid metal complexes using special transporters that they have. And I'll show that process. Microbes also will take nutrients from other microbes. Okay, and how do they do that? They make what are called biosurfactants. And those biosurfactants make membranes leak. So a microbe, like for example, bacillus, will produce surfactant and make membranes of other bacteria and fungi leaky. And then they can then acquire those nutrients. So there's, we think that's part of this phenomenon. Siderophores, okay, how do they work? There's a special molecule here that the microbes produce, this little blue thing here, that's the siderophore. Here they put that out, send that out, it attaches to an iron, for example, and then it will carry that back and get into a receptor, and then the microbe will absorb it in. Okay, the cell walls of microbes have a negative charge, and the iron will just stick on it. Metals will stick on it. Zinc will just stick on it. This actually is a bacterium, and it shows all these little silver dots. That's silver particles sticking to the wall. So when the plant takes the wall away, it gets microbes right there. It carries microbes. Okay, the other process is we know, and I think this is getting to the question that you asked a few minutes ago. We know that plants secrete organic acids into the soil, citrate, acetic acid, for example, citric acid, malic acid. And those, we also know that those organic acids will go out and attach to metals, iron, zinc, and so forth, attach to those metals and complex, just cover them over. Microbes have these organic acid metal complex transporters. So the microbes can go find those complex metals and then hook onto them and suck them in. So, and then once they get them, they can then travel back to the plant and re-enter the plant where it can be harvested. So this is a kind of a system where the plant is actively working with the microbe to maximize nutrient absorption. Okay, and this just shows details of some of that. The microbe can use the citrate as an energy source, so it actually is rewarded for doing that. What happens to plants without the rhizophagy cycle? Okay, so this is a rice, and we removed all the microbes and we put a pseudomonad in it. And you see these big seedlings, and these have no microbes at all, and you can see very tiny seedling development. A little bit older seedlings, you can see here. Here, this is with the microbes, with the Pseudomonas. You can see the plants are bigger, maybe about 30% bigger. And you also see the soil adhering here. So the relationship with the soil, the root hairs and that relationship sticking the soil to the roots is more significant here than you have here without the microbes. So it does appear to be significant. Okay, the very last thing I'm going to mention is that the rhizophagy cycle may be affected by carbon dioxide levels. And the reason for that is that carbon dioxide in the air will suppress reactive oxygen in the plant. And I'm sure you've all heard that There's some work going on looking at nutrients like zinc and iron in crops, decreasing in these carbon dioxide enrichment experiments where they expose plants to carbon dioxide and then measure nutrients going into the plant. So it's suppressing those nutrients. And that's a few years now people have been looking at that. Well, as it turns out, we did some experiments to evaluate, whether high CO2 is impacting the reactive oxygen. And this just shows this is at current levels of CO2 in the air. And you see it's a bit darker. This is in an experiment with a bit higher. This is like the 50 to 100 year level. So it's a little less. So the levels of reactive oxygen appear less. We actually, to measure this more precisely, what we did is we can grow plants, remove all the microbes, grow the plants in protein, and protein will stimulate reactive oxygen formation. And then we do that different CO2 level experiments. And then stain it for reactive oxygen. You can see all this brown, the roots are in there. You can see at the current levels of CO2 in the atmosphere, you see there's kind of thick, broad zones around those root tips. But this is in the higher level, the 50 to 100 level. You see they're very fine, very fine. There's less reactive oxygen around those tips. So we don't know if that means, that plants, that this is just destructive, this climate change is going to be destructive to the rhizophagy cycle. Plants might be able to adapt. We just don't know that yet. And we don't know, this actually has to be confirmed somewhat. What is clear is that we want to conserve this underground biology that's happening with these microbes. And what that means is, for one thing, there's on seeds, so don't use seed treatments. Anything that sterilizes the seed or interferes with microbes on the seeds, don't remove those. Organic amendments to the soil, increasing organic matter is important. No till, don't disrupt, don't churn the soil so that you disrupt those microbial community, and so forth. So that's really all I have to say.
Thanks for listening to these conversations today. We love bringing our listeners research that helps guide our understanding and adoption of practices that help us mimic nature. And as always, if you'd like to learn more about what we're doing to help growers implement great soil health practices, check out our website at asn.farm, and there you can click on links to follow us on Facebook, Twitter, Instagram, LinkedIn, and YouTube. There's a lot of great things happening and always something to learn. Thanks for listening.